Good morning to all. In this session, we will see how to find the map distance between two linked genes. Distance between two linked genes is usually measured by map unit. 1% crossing over between two linked genes is known as 1 map unit or 1 centimorgan. Map distance between two linked genes can be measured using a formula. Map distance or recombinant frequency is equal to number of recombinant offspring divided by total number of offspring into 100, where 1% recombinant frequency is equal to 1 centimorgan or 1 map unit. First question was asked in CSIR NET exam December 2012. A cross was made between pure wild-type males and brown-eyed curl-winged females of Drosophila melanogaster. The F1 female were test crossed and F2 progeny are given here. Now we have to find out the distance between brown-eyed curl-wing. In the question they have given brown-eyed curl-wing female, we'll consider that as capital BW and wild-type male as small bw. Now the cross was made between female and male. The F1 progeny were obtained to be a brown-eyed curl-wings. Now this one is crossed with the wild-type male and F2 progenies are obtained. That was given in the question. Now what we have to do is we have to compare the genotype of the parent with the offspring. Now if you see the 30, these two are the recombinant offspring. The genotype of this offspring is not similar to the parent offspring. Now we have to use this 30 and 20 for finding out the recombinant frequency. Now the map distance between the gene A and B is equal to number of recombinant offspring divided by total number of offspring into 100. So here the number of offsprings are 30 plus 20 divided by 400 into 100, that is 12.5. So the map distance between the brown eye and curl wing is 12.5% or 12.5 centimorg. So the answer is 1. The second question, this question was asked in CSIR NET June 2016. Poplar is a dioecious plant. A wild plant with genes capital ABC was crossed with triple recessive mutant small abc. F1 male hybrid was then back crossed with the triple mutant and the phenotypes are given. Now what we have to do is we have to find out the map distance between A and B gene and B and C gene. In the question they have given homozygous capital ABC is crossed with the mutant small abc and the F1 progeny obtained are a heterozygous. Now the F1 is crossed with back crossed, that is back crossed with the recessive parent. So they have given the number of progeny after the cross. Now what we have to do is we have to compare the genotype of the progeny offspring with the parent. Here the parent we have to compare the genotype of the offspring. Now in order to find out the map distance or recombinant frequency between A and B gene, compare the genotype of the offspring with the parent. Consider only the A and B gene of the offspring and the parent and find out the offspring which is not having the similar phenotype with the parent. So in this case this 4 are considered as the recombinant offspring because it is not having the similar phenotype of the parent. So the recombinant frequency between A and B equal to 14 plus 120 plus 100 plus 16 divided by 1000 into 100. So the recombinant map distance between recombinant frequency between A and B is equal to 25% or 25 map unit. Similarly, to find out the map distance between B and C, we'll consider the B gene and C gene of the parent and that is compared with the offspring genotype. Now in this case, the recombinant offsprings are this 4. Right? So the recombinant frequency is equal to 65 plus 14 plus 16 plus 75 divided by total number of offspring is 1000 into 100. So the recombinant frequency, the map distance between B and C is equal to 17% or 17 map unit. So the answer is first one, 25 plus 17, or the map distance between A and AB, BC. So in this session we have seen how to find the distance between two genes. In the next sessions we will see few more genetic related problems. Thank you.